I would like to begin thanking Jim. I don't know if I thank you or or not thanking you about inviting me here, but thank you for the invitation. Actually the video that we just saw, I think it's a good introduction of my talk because I'm gonna be talking about biology process, and if you mind, I prepared a presentation of the work that we do in the laboratory in Spain using the human body as a factory for treating coronavirus infection. So I'm gonna present a little introduction of the structure of the virus, and then I'm gonna tell you how the immune response is working for this virus, and then what we have done to use that immune response as a tool, the technology we use to use that immune response as a tool for treating the infection. So in the first slide, you can see the structure of the virus, and believe it or not, this virus is really simple compared to other human viruses. It has a really small genome that codes for not a high number of proteins. Some of them are in the slide, such as the spike protein, that is the one that have been used to develop the vaccines, and other membrane or nuclear proteins, such as protein M or N, that I will talk about them later during my talk. So in the next slide, I show how the virus connect to the cells. So the virus actually is called coronavirus because corona is the Spanish word for crown. And as you see, the first time that this virus was seen under the microscope, it shows like it has a crown. So the crown has this spike that is the S protein that is actually work connecting to the cellular receptor that is shown in the slide. And this interaction in between the S protein, the spike protein, and the cellular receptor um, allows the virus to enter in the cell and replicate to make more viruses that can be spread to other cells in the body. So in the next slide, you see how the immune response is created against this virus. So when the virus infects the human body, because it's a respiratory infection, usually infect the epithelium that is located in the mucosa, in the nose or in the lungs or in the eyes, and it disseminates to other cell types. When this happened, the body responds with an immune response because recognize the virus as a strange, and use different cells that are called T cells to fight against this virus, to elicit an immune response against the virus. So some of these cells produce antibodies, which are, some of them are neutralizing antibodies that recognize this spike protein and bind to the spike protein and block the infection to other cells. That's why it's called neutralizing antibodies. But there is other kind of T cells that are able, well, the neutralizing antibodies are able to block the infection of the free virus. It's viruses that are not inside of the cells and are looking for other cells to infect. But there is other T cells that are specialized to recognize the infected cells. So it recognizes the virus that is inside of that cell and kill those cells. And those are the one that I point out in the bottom part of the slide. So in the next slide, you can see how if you block through neutralizing antibiotics, you block the spike protein in the surface of the virus, you actually block the infection of new cells. But then we ask, in the next slide, we ask ourselves, can we transfer, can we focus on, this is the neutralizing antibodies, the response that is being used for developing vaccines. But well, can we use the T cell response that actually recognize this infected cells from convalescent patients or patients that already had infection and have elicited an immune response to treat critical patients? So the goal here will be to use these T cells that are specific, that specifically recognize the virus once a person has the infection, to extract those cells and use them as a treatment. So how we do that? Well, we need a donor, which is a person who already had coronavirus and had already a positive immune response against the coronavirus. And we take the blood, and through a process called apheresis, we take some of the components of the blood and put the blood back in the patient, of course. And this part of these components of the blood is the T cells. So we actually use those T cells to treat patients. How we do that? So in the next slide, I am showing you the technology we use. So when you get from the apheresis, you get a mixture of T cells that are against multiple pathogens. But we want to separate those that are specific only from coronavirus. So how do we do that? Well, we had to stimulate those cells with proteins of the virus. And in this case, we use actually the proteins I told you in the first slide, that is the S protein, which is already known that elicit an immune response. But we also use the M protein and the N protein as a stimulus to activate these immune cells. So when you stimulate those cells, the cells actually produce a molecule that you can detect. And through the recognition of that molecule, you can separate the population of the cells that actually are activated against this specific stimulus and are specific for coronavirus. So we separate those cells and propagate them in the laboratory to make more cells, because we need a lot of cells to treat a patient. And then we tested if those cells that we separated actually recognize infected cells and kill them. And we actually do a protocol in the laboratory with infected cells and mix those infected cells with the cells that we isolate and check if the infected cells are dying. And actually we did. So we discovered actually that of the 3 proteins that we use for stimulation, the M protein is the protein that is the best to stimulate this kind of response. And all of them does, but M protein is the one that stimulates more number of cells and have a higher effect on infected cells. So in the next slide, which is the last one, I just want to show you how we can use this. So we have, as you know, a huge number of people in the world that have been infected by coronavirus that are actually immune to the infection. This immunity probably doesn't block new infections, but decrease the effect of the new infections. So holding somehow the infection and not producing the side effect that we had in the previous wave. So with this multiple number of people, we can actually extract the blood and use that blood to do the process that we did in the laboratory and make frozen cell banks that are there waiting for someone that need it. So if you have those frozen cell banks in the hospitals, when someone in critical care that doesn't have the immune response or that is not working as fast as it should, you can actually transfuse those cells to the person, those cells that are specific against coronavirus through intravenous administration in this hospitalized patient, and actually transfer the immunity from a person who already have immunity to a person who doesn't have immunity and can be fighting the virus with the donor's immunity. And that's it. I would like to finish in the last slide just thanking everyone for your attention, and also the people of my lab that is been working in this project. I think this is one more slide. Yeah, this is my group in Spain. And thank you for your attention and the group of the people in my lab. Thanks.